So you use the term synthetic immunology or synthetic biology. Could you explain a little more about what that is?
We know a lot about how immune cells function. So we know a lot of the receptors that activate them. We know the downstream sort of signal transduction cascades that are necessary for regulating immune cells. Synthetic biology is kind of where you go in and sort of hijack these systems for your own purposes. Now we can go in and modify cells with new genes, or we can take pieces of different proteins that we know are functional in, let's say, a T cell, and start to repurpose them to where now we can build cells that have new synthetic receptors that can detect different aspects of tumors, maybe could detect certain tissues, detect certain autoinflammatory sites. But instead of just simply activating these cells in these microenvironments, they really start to sculpt and regulate that cell behavior in such a way that it's highly controllable. And that's the only way we're going to be able to, utilize cell therapies in a vast array of diseases, because it is something that could be quite toxic and unwieldy. But if you have complete engineered control over these cells and how they function in the body, then I think they'll be quite useful and quite effective in the future.
So one of the big challenges that we have in the solid tumor space is really this tumor microenvironment, which is highly immunosuppressive. Could you talk a little bit about how you're using the approaches of synthetic biology to overcome some of those suppressive effects?
I'm excited to be part of institutes like the Parker Institute, where they're breaking down cancer from all these different perspectives. So we want to figure out how to target the cancer. We want to understand the different microenvironments that exist across all these different cancer types, also across patients within a single cancer type. So once we have some of that information of what are the major suppressive mechanisms that are occurring in that solid tumor, we do have platforms such as the synthetic Notch receptors to really control the cellular functions. These cell therapies can resist these sort of recalcitrant environments that exist within tumors. When a T cell goes in, it activates, but then over time, it becomes inert and can no longer activate. I think we know enough about some of these processes that occur in T cells or other immune cell types to now start to engineer mechanisms within these cells to resist these kind of deleterious effects that the tumor imposes upon the immune system.
One of the advantages of using the immune system to fight cancer is you get this long-term persistence that you see with natural immune responses. How are you using synthetic biology to leverage that or to improve that persistence?
One of the beautiful aspects of our immune system is what's called memory. And so if you're infected by a pathogen, that first response now leaves memory cells that if you're reinfected, you have cells on board that can rapidly respond to disease. So instead of being sick for a week to 10 days, you're only sick for a few days, et cetera. So we have spent decades studying these sort of processes. So now we can start to modify the receptors that we put into these cells such that they drive this sort of memory phenotype. With some of the platforms we built, we can control genetic circuits that are upregulated in these T cells in the microenvironment of the tumor such that you can drive genes that will lead to persistence of the cells. Tumor therapy now is kind of immune metabolism, so controlling the metabolism in these T cells. Pretty much all of the platforms that we're building to engineer into these cells can be used to control these different processes.
So could you highlight some areas in which synthetic biology has improved or is improving the efficacy and safety of cancer immunotherapy?
We've actually built receptors that instead of when they see the tumor, they can't activate just from that single input. So the way we build the receptors is you have to titrate in small molecule sort of drugs in order to give that cell a second signal to activate. And then the idea there is that we can infuse these sorts of T cells into a patient and then titrate in a drug and regulate the activity of these cells such that maybe you reduce the chances of adverse events in patients. patients, but you still get the efficacy. More recently, we're actually building totally new receptor platforms for cells. We call them synthetic notch receptors, but they're really a platform for many different cell types to recognize a site of disease or a tissue in the body, and then very precisely perform therapeutic functions. So instead of a cell doing many different things, we have cells do very specific functionalities, and we're starting to understand some of those things that are causing toxicities at this point, and we're trying to modify cells such that they don't do that.
Do you think that these aspects of synthetic biology are really going to be implemented into all the different cell therapies?
I think over the next 10 years, what I hope to see is that we're not simply utilizing cells for catastrophic cancers, where it's late-stage metastatic disease. No one with autoimmune disease that can be treated with other sorts of therapies that are potentially less dangerous and less toxic will want to have a cell therapy. So I think synthetic biology is the answer. If we can build control systems into these cells where they can titrate their behavior in the patient, then we can go into these sort of disease scenarios without worrying about having terrible repercussions where you have major issues in patients.
With the level of control that you're bringing with this synthetic biology, that you may be able to begin to build cells that work well with even standard of care chemotherapy. They have cells that are resistant to chemotherapy such that you can get an anti-tumor effect directly from the chemotherapy and still have the cells persisting and functioning. Are you aware of research in that field that's really kind of looking at what standard of care is and how can we build cells that function well in standard of care?
One thing I think about is that people go through all these pre-treatment regimens. Almost anyone that gets cell therapy has had many treatments beforehand. One example about is that there's certain lung cancer treatments in which after you have these sort of therapies, the antigen landscape on these tumors changes. And then you can start to think about how can you target your cell therapies to tumors that have already been treated in certain ways. You can develop a treatment regimen such that you push that tumor into a certain place where it's more easily eradicated by cell therapy or easily targeted.
We can envision a future, at least maybe even in the near future, where synthetic biology and the tools that you're developing can turn the metastatic cancer into really a manageable chronic disease where we can monitor what's going on and then treat appropriately with the right kind of cell or the right kind of therapy.
I think what's great about cell therapies is that most cells that we're engineering are built to get into these places in the body, right? So when you have massive metastatic disease or solid tumors, we'll get to the place where it's much better than using a biologic or small molecule. First of all, the cells can penetrate better. And the second thing is cells are smart robots in which they can go in and make higher order decisions that can evolve over time in the patient. So the cell's the one that can really control the therapeutics.
Sure, the autonomous agents of change of the immune system. Right. This is really what cells are.